Welcome to The Explainer. Today, we're diving into a document that is, no exaggeration, a make or break deal for the future of medicine. We're talking about the Saudi Food and Drug Authority's blueprint for getting gene therapies approved. So let's figure out what it really takes to get from the lab bench to the patient's bedside. So let's just jump right in with a question. What do you think is the #1 reason a really promising gene therapy trial gets shut down by regulators? You'd probably guess it's something to do with the science, right? Or maybe the early clinical data didn't look so hot. But nope. The real killer is often something, well, a lot more fundamental.
The answer is CMC. Chemistry, manufacturing, and controls. That's it. It's not about if your science is brilliant. It's about can you make it? Can you make it safely, consistently, the exact same way every single time? Because if you can't, even the most groundbreaking therapy is dead in the water.
Okay, so here's our game plan. We're going to start with what I'm calling the CMC gauntlet. Then we'll get into the nitty-gritty of crafting the drug substance and the drug product. After that, a deep dive into the unique challenges of CAR T cells. And finally, we'll wrap it all up with the most important takeaways you need to know. Let's do this. All right, Section 1, the CMC gauntlet. You know, before we can even begin to talk about manufacturing, we all have to be speaking the same language, the regulator's language. So let's lock in the 3 key terms that are the absolute foundation for any submission. First, we've got the drug substance, or DS for short. Think of it like this. If you're baking a cake, the DS is your pure, concentrated flour and sugar. It's the raw, powerful, active ingredient of your therapy before you've done anything else to it. Next up, the drug product, or DP. So if the DS was the flour and sugar, the DP is the fully baked, frosted cake ready to go. It's the drug substance mixed with all the other inactive ingredients, we call them excipients, that make up the final medicine that actually gets administered to a patient. And last but definitely not least, we have critical quality attributes, or CQAs. So sticking with our cake, CQAs are the checks you run to make sure it's perfect. Is it the right size, the right texture? Is it sweet enough? For a therapy, these are the non-negotiable characteristics, things like purity, identity, and potency, that regulators are going to scrutinize to make 100% sure your product is safe and effective batch after batch. Okay, so now that we have the language down, let's get into the how. Section 2 is all about crafting that drug substance. We're going from the very first raw materials all the way to a purified final product. So what does this actually look like? Well, think of it as a sophisticated biological assembly line. You start by growing up huge batches of cells. Then you use a vector to get the genetic material inside them. From there, it's all about harvesting and then a whole series of purification steps to get rid of everything you don't want until you're left with that pure, concentrated, active ingredient. Okay, let's talk about starting materials, because I promise you, regulators are absolutely obsessed with them, and for very good reason. Where you get your raw ingredients from defines your whole risk profile. So if you're using animal-derived products, you're immediately worrying about things like TSC, mad cow disease. But if you go with human-derived materials, the big worry shifts to human viruses like HIV or hepatitis. This choice, right at the beginning, sets the stage for your entire safety plan. So how do you keep things consistent? The SFDA, like most regulators, points to a 2-tier banking system. It's a really smart approach. You create one pristine, heavily tested master bank. This is your golden source. Then for your day-to-day production, you create a working bank that's derived from that master. This whole system guarantees that every single batch you ever make starts from the exact same, qualified, and safe place. Okay, time for a deep dive on something that keeps developers and regulators up at night. It's the hidden danger of what we call replication-competent viruses, or RCVs. See, most gene therapies use a viral vector that's been engineered specifically so it can't make copies of itself. The nightmare scenario is that during manufacturing, some of those vectors could mutate or recombine and revert back to a version that can replicate freely inside a patient, and that is a massive safety problem. And just look at how incredibly strict the SFDA's limits are here. For something like an adenovirus vector, the acceptance limit is one replicating particle for every 30 billion viral particles. That is an insanely low number. For other vectors, you have to test at multiple points in your process. The message from regulators is crystal clear. This is a risk you have to eliminate, period. All right, so we've made our pure drug substance. Now we've got to turn it into a medicine. Let's move on to formulating the drug product. I want to pull a quote directly from the guideline here because it's so important.
It says, a lack of understanding of in-use and in-device stability may not adequately justify the risks associated with clinical study treatments. I mean, that's the SFDA basically putting up a giant red flag. They are telling you exactly where people mess up.
And this is the exact detail that trips up so many companies. Your therapy can be perfectly stable in its glass vial, but what happens the moment it touches the plastic of an IV bag or the inside of a syringe? Does it stick to the surface? Does something in the plastic actually degrade it and reduce its potency? You absolutely have to prove that your drug is stable and active, not just in the vial, but all the way through the delivery device and into the patient.
Okay, it's time for a masterclass. Let's take everything we've talked about, drug substance, drug product, all the controls, and apply it to one of the most complex and personalized therapies out there, CAR T cells. Right off the bat, CAR T presents you with this huge dilemma. Do you go autologous, where you use the patient's own cells? The big win there is you don't have to worry about immune rejection. The massive challenge? Every single patient is a brand new, unique manufacturing run. Or do you go allogeneic and create an off-the-shelf product from a healthy donor? That makes manufacturing way more consistent, but now you have a whole new set of problems, like intense donor screening and the risk of the patient's body rejecting the cells. So if you're going the autologous route, your single biggest challenge is taming that incredible patient-to-patient variability. Your starting material is different every single time. So the only way to get a consistent final product is to have an absolutely rock-solid, lockdown manufacturing process.
So how on earth do you control for that kind of chaos? Well, the SFDA guidance is clear. You lock down your process parameters. For example, you use a fixed multiplicity of infection. That's just a fancy way of saying a very specific calculated ratio of virus to cells when you're doing the gene transfer. You use a fixed B to cell ratio for activation. You standardize everything you possibly can. The goal is to make the process the constant, even when the input is variable. And of course, a strict chain of identity is absolutely critical to make sure patient A's cells go back to patient A and nobody else.
Okay, here's a super important and subtle safety detail. When you're measuring something called the vector copy number, which is basically how many copies of the new gene get into the cell's own DNA, the SFDA is very specific. They say you have to calculate that number based on the cells that are actually CAR positive, not just the total number of cells in the bag. Why? Because that gives you the true, most accurate picture of the potential safety risk. All right, we're in the home stretch. Let's talk about what happens when you need to change your process, and then let's boil this entire guideline down to the absolute must-know takeaways.
Okay, if you walk away from this with nothing else, remember these 5 things. This is your core CMC checklist. 1. Safety from adventitious agents. That means rigorous testing and sourcing for all your raw materials. 2. In-use compatibility. You have to prove your product works in the final delivery device. 3. For CAR T, control that variability with fixed process parameters. 4. Testing for those replication-competent viruses is absolutely non-negotiable. And 5. Retain samples. Keep material from every single batch, because you're going to need it later if you ever change your process.
And that really leaves us with a pretty fascinating question for the future, right? As these therapies get even more advanced, we're talking about in vivo treatments that happen inside the body, or CRISPR-based CAR T. How are these fundamental CMC principles going to adapt? I mean, how do you ensure quality and safety for a batch of one that's manufactured inside the patient themselves? That is the next great challenge for this entire field.